The molecular and cellular mechanisms that lead to heart failure in people with cardiomyopathy are determined by the specific gene variant that each patient carries, according to newly published research based on the first comprehensive single-cell analysis of cardiac cells from healthy and failing. The work, reported in the journal Science, was conducted by 53 scientists from 6 countries in North America, Europe, and Asia. The study shows that cell type compositions and gene activation profiles change according to the genetic variants. The investigators say the findings can inform the design of targeted therapies that take into account each patient's underlying gene defect responsible for their particular form of cardiomyopathy. The team studied 880,000 single heart cells, examining the genes activated in about 880,000 single cells from 61 failing hearts and 18 healthy donor hearts. As reference was a complex endeavor which required an interdisciplinary team. The organs were procured by the Brigham and Women's Hospital in Boston, USA, University of Alberta in Canada, the Heart and Diabetes Center North Rhine-Westphalia in Bad Oeynhausen, Ruhr University Bochum in Germany and Imperial College London, UK. Joint first authors are Dr. Daniel Reichert, Harvard, Eric Lindberg and Dr. Henrique Motz, both MDC. A disease with many causes. The scientists focused on dilated cardiomyopathy, DCM, the most common form of heart failure that leads to heart transplantations. It involves an expansion, dilation, of walls of the heart chamber, especially in the left ventricle, the heart's main pumping chamber. The muscles of the heart become weakened, compromising its ability to contract and pump blood, which ultimately leads to heart failure. The consortium studied tissues from patients with different genetic mutations that commonly lead to cardiomyopathies. To our knowledge, this is the first such analysis conducted in cardiac tissue, and we hope this approach can be used to study other types of genetic heart diseases. The scientists precisely characterized the various mutations in each of the hearts and compared them with each other, as well as with. No lab on its own could deal with the massive amount of data generated, but close collaboration among specialists from different disciplines made it possible to assemble a coherent picture out of each individual piece of the puzzle. This study is also part of the efforts of the International Human Cell Atlas, HCA, consortium, which is aiming to map every cell type in the human body as a basis for both understanding human health and for diagnosing, monitoring, and treating disease. Only this level of resolution allows us to see that cardiomyopathies do not uniformly trigger the same pathological pathways, says co-senior author Christine Seidman. The number of these cells stays the same. But the existing cells become more active and produce more extracellular matrix, which fills in the space between the connective tissue cells, Eric Lindberg adds. Thus, rather than overproduction of fibrotic cells, the researchers observed only a shift in the proportion cell subtypes, marked by an increase in the number of fibroblasts specializing in the production of extracellular matrix. The phenomenon was especially pronounced in the hearts of patients with a mutated RBM20 gene, Henrique Motz explains. This observation was also reflected in the patients' medical histories. On average, patients with this specific mutation suffered heart failure and needed a transplant much earlier than people with other genetic forms of DCM. Although this form of cardiomyopathy can also be caused by a mutation in several genes, the team focused its analysis on the gene for the protein PLAKOPHILIN2, or PKP2 for short. The team found that different genetic causes of cardiomyopathies were associated with specific aberrations of the cellular communication networks. This is clear evidence of specified mechanisms driving the disease. Finally, the scientists used artificial intelligence to develop a model from all these data. Based on the specific patterns of molecular changes in the various cell types, the algorithm can predict with high degree of confidence which mutation is present, confirming that differences in gene and cell activation are associated with pathogenic variants of specific genes. The consortium has made all of its results available to the scientific community online. Seidman hopes that this resource propels studies by other groups to define new treatments that prevent heart failure, which today is an incurable disease. We hope that future pharmacological treatments will at least slow down the progression of the disease, and that the data from our study will help make this happen. Meanwhile, the research team has identified its next task. The heart tissue that we studied came from people in the final stage of a disease, Daniel Reichert, one of the first authors, says. We are excited to see what changes we discover in earlier stages of disease, for example based on endomyocardial biopsies. Perhaps biomarkers and pathways will be found that elucidate a very precise disease pathogenesis, truly enabling personalized medicine, Gavin Udit adds. Make a difference. Sponsored opportunity. StorySource. Materials provided by Max Delbruck Center for Molecular Medicine in the Helmholtz Association. Note: content may be edited for style and length. 2022, August 4. How pathogenic gene variants lead to heart failure. Science Daily. Retrieved August 5, 2022 from ift.tt/9axnot Max Delbruck Center for Molecular Medicine in the Helmholtz Association. Thanks for watching. Please subscribe my channel for more videos.